And as the second speaker, I would like to introduce to you all Dr. Preetha Balakrishnan, who is a scientist with a very interdisciplinary background. So she has some experience in biomodeling and in silico analysis, also in clinical interpretation and reporting of rare diseases. She did a PhD in molecular cell biology in the University Hospital of Vienna, and then also worked in digital healthcare data analysis and scientific writing. And last year she joined us at Limbus Medical Technologies. We're very happy that you joined us, also joining us here today. Welcome, Preetha.
Thank you, Yvonne, for this kind introduction. Good afternoon, everyone. And I would take the next 15 minutes, hopefully, to enlighten you about T2T and what it can do and how it can help clinical diagnostics. So, when we get a patient sample and when we sequence it, what we get out of the sequencer are reads which are not aligned. And for aligning it, normally we use a reference genome where then we can compare it with the patient sample and we can call the variants out. But one also has a choice sometimes to do the alignment de novo. But it is compute intensive, time consuming, and it also lacks annotation. On the other hand, if we use a reference genome, it is efficient and it is cost effective, and what we know that we have a lot of annotation sources because of which we can compare and call out variants which is less error prone. This says that we really need a reference genome which is complete, accurate, and it also has a high quality. And we know that scientists in our field has strived to achieve this for a long period of time. What we see is the UCSC releases currently. In 2000-2001 range came the first complete draft of the human genome sequence, hg38. It had 2.6 gigabase pairs, and it still had 10 to 15% of gaps. 9 years later, we had hg38, which had 2.8 gigabase pairs totally, and it had still 5% of gaps which were not represented. Then we had hg38, what we all know, it's our current standard. It has 4 to 5% gaps. And now, in 2022, with the long-read sequencing technology coming in, we now have HS1, which is our first gapless assembly, where the telomere to the telomere, there is a complete representation of the genome. And this methodology has now been adopted by the Pangenomic Reference Consortium. And now, as of 2023, we have 47 haploid assemblies. So what do we know today about the T2T? HS1, which is nothing but the T2T CHM13 assembly, comes from CHM13 human TERT cell lines, which are immortalized cell lines, which are derived from the homozygous complete hyatidyl mole. That means to say it is an abnormal condition where an egg without a nucleus is fertilized by the sperm, and now the paternal chromosome gets duplicated, as a result of which we have a 46XX. And therefore, we do not have a Y chromosome. And that is basically done by using hg002 sample, that is genome in a bottle sample, and the Y chromosome comes in, and these combined together is now called as the human sequence 1 by UCSC. To study this T2T, they use sequencing, the authors of this paper use sequencing technologies of PacBio, Oxford Nanome4, and Illumina. And assembly was done with Vinomab2, BWA, and the filtering was done with Merfin. We learn that T2T adds or revises 229 million base pairs of sequences. Where does this come from? It's mainly in the centromeric regions of chromosomes or in the short arm of acrocentric chromosomes. We also have some telomeric regions, and a large portion of it is also on the Y chromosomes. When we talk about it in a region-specific manner, where does it come? It is mainly in the centromeric satellite regions. It is also affecting segmental duplications. And what we also see is part of it also goes to the rDNA, which was also previously not resolved. So what does it mean at the gene level for us? We have 5.8% genes and 0.6% of protein coding genes which have increased compared to hg38. And in terms of transcripts, we have found 2.5% protein coding transcripts which have increased. And of note here is also that the issues which were there previously in hg38 has been reduced to about 96.5% in terms of million base pairs in the T2T. So how does this affect variant calling for us? If we consider long-read sequencing, what we can see here is the mismatch rate has reduced, which is represented in orange for T2T, and blue is hg38. We can see a reduction irrespective of the technology you choose, that is PacBio or Oxford Nanopore. We also see that the coverage standard deviation has highly reduced, as a result of which the reads are more homogeneous in coverage and there is less noise. As a result, you have the structural variant calling, which is better, and you can also see some previously unresolved regions. And here you also see that the deletion insertion balance is much better compared to that of hg38. So does it also affect short read sequencing similarly? Yes, it does. So we see that the mismatch rate here compared to that of hg38 is much lesser across the population. And it is also similar in terms of coverage and standard deviation, as a result of which the SNV calling and the indels are much better, and you can also see some previously unresolved regions or the SNPs which are called. So yes, we see this, but there has to be some kind of validation, some kind of benchmarking done as well for us to be able to use it. So this was done by taking 273 medically challenging genes which have been reported. They took it from hg002 and then they matched it back to T2T, took only those genes which corresponded to that of hg38, and then they continued that in hg002 by using 3 different technologies: Illumina, PacBio, and Oxford Nanopore. When you compare this, we observe that the false negatives as well as the false positive rates were considerably reduced. That means that the false calling is less and we can be more confident when we are reporting cases. So in total, there were approximately 622 medically relevant genes that were impacted by T2T. So I think it's better that we see at least a couple of examples to know what it means to us in the clinical sense. So here we have the first example where we're considering a false negative duplication. This means that there actually is a duplication, but we do not see that in hg38. So we're considering an example of this gene, KCNJ18, and it is implicated in thyrotoxin periodic paralysis 2. What we see here is that KCNJ18, which is there in chromosome 17, and it has a segmental duplication region. The top panel is Illumina, the central panel is PacBio, and the last panel is Oxford Nanopore. In all cases, we see a large coverage, but we also see that the number of variants which are called, because of the large number of colors which you can see, is extremely high. This is happening because of the absence of a duplicated region, which is supposed to be in KCNJ17, which has now been resolved by T2T. We can now see that there is less number of variants which are called, and the coverage is also adjusted. So we have a false negative case. Now we can look at a false positive case. So what happens in case of a false positive duplication? Here is an example of KCNE1. It is also another potassium channel, and it is implicated mainly in cardiac diseases like long QT syndrome. And here we see that in hg38, there is less number of reads in the Illumina, in PacBio, or the Oxford Nanopore panel. And this is mainly because of a false duplication. The reads go and match also to the other region, which is there in KCNEB1. And this has also been corrected in T2T, as a result of which we can now see a complete coverage, and there is some amount of variant calling. So we can now see a case study of how this has been represented. We are talking about Kleefstra syndrome 1, where the proband was a child who came in with the symptoms associated with Kleefstra syndrome, but they noticed that the parents were unaffected. That means to say that it was a de novo variation. What they observed when they karyotyped was that there was an inversion in chromosome 9 between Q12 and Q34.3. But they could not observe this breakpoint in hg38 or hg19. And what the breakpoint did actually was it disrupted intron 25 of this EHMT1 gene, which leads to haploinsufficiency, as a result of which it caused the syndrome. What we see here in T2T is that in long-read sequencing, they could observe these breakpoints, which we can see here, which I've depicted here in the yellow box. This was not clearly visible in short-read sequencing, but they could do this with long-read and with T2T, and they could resolve and see this better, as a result of which it could help in now validating the case as well. So yes, T2T is pretty promising, and we do see differences which can help us in the clinical diagnostics. But when can it be the new standard? Well, for something to be a new standard, it is very important that we can address some points. The first of which is quality. And we know, we already saw previously that there is very good quality. And in 2022, it had a quality of 73.9. And currently, the T2T is really striving hard to come up to Q100. I don't know if that's possible, but they do. Then they thought about benchmarking as well. Yes, we saw benchmarking, but we need to know that it has not been published yet in the GIAB website. So it's still ongoing. Talking about diversity, T2T still lacks diversity because it's a haploid set, because it was just the paternal chromosome which came in. We do not have the maternal complement. But I think in this sense, the pangenome can help, because here in 2025 spring, they claimed that they would have at least greater than 200 samples. But we need to know that it is a graph-based approach, so to use it would also be slightly harder, and we need upgraded tools and technologies for it. And we need annotation sources. Currently, UCSC and RevSeq have T2T in them, but the rest of the sources, we did a small curation, and we looked at it, and we found that not many tools are supporting T2T currently, but CAD, DBSNP, and GenCode have sort of started doing it, and we can see it in the near future. But for the rest of the tools, it would take some more time for it to come. So in summary, we can say that T2T covers previously unresolved regions like segmental duplications, centromeric regions, and short arm of the acrocentric chromosomes and most parts of the Y chromosome. 622 genes which were medically relevant were impacted from this assembly. We did a small dig into our internal system, internal reference database called Alexis, and we found that approximately 465 of these 600 medically relevant genes were reported, and it was approximately 30% of the cases. So it could be that 30% of the cases are impacted. T2T is definitely promising, but of course, like this dog, we need to wait for a few more years for it to become the golden standard. Thank you.